So just like people do, cells produce garbage or waste. And the cells have to be able to get rid of that waste. So this waste might be some sort of metabolic byproduct from the cell that the cell can't use, or it might be old worn out proteins or old worn out mitochondria. Um, or the cell might also need to digest food, begin the breakdown of food that the cell has brought in. In order to do either of those things, the cells have an organism called the lysosome. So, our book says that the lysosomes are basically garbage disposals. I kind of prefer to think of them as stomachs, although it really depends on what process they're actually doing. So the lysosome is filled with up to 50 different digestive enzymes, and they're often acidic. So just like your stomach is acidic and has molecules that aid in your digestion, the lysosome does too. So the lysosome works by either fusing with a vesicle, say one from phagocytosis, and sort of spilling its digestive enzymes into that new vesicle, or it can also engulf a degraded mitochondria. So after about 10 days of intense activity, the mitochondria is done and it needs to be broken down and a new one produced. White blood cells need to be broken down because they are constantly battling bacterial invaders, and so we don't want that staying around in our system. So that's where the lysosome comes in. The lysosome is going to break down whatever product is on the inside, whether it's waste or a food product that's been brought in, and it's going to do that via those digestive enzymes. There's a disease called Tay-Sachs, which is a fatal accumulation of lipids in the brain. Now, this disease comes from the lack of a specific lipid digesting enzyme found within the lysosomes. So Tay-Sachs is an inherited disorder, and due to the dysfunctional gene, just like cystic fibrosis, individuals with this disorder accumulate lipids in their brain, and it eventually interferes with normal function and is usually fatal by age 2 or so. So let's move on to a network of membranes within the cell called the endomembrane system. Now, the endomembrane system is simply a system of membranes inside the cell. So the endomembrane system is made up of 3 major components: the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and the Golgi apparatus. Now, the overall function of this endomembrane system is to produce and modify molecules, and those molecules are either going to be exported elsewhere in the cell, or they might be exported for use outside of the cell. The endomembrane system is also important in breaking down cellular byproducts and also toxic molecules that might have entered the cell. The rough ER gets its name from these bumps on the outside, and these bumps are ribosomes. So remember I said earlier that ribosomes are the site of protein production. So a little bit of genetic information will leave the nucleus and move to the ribosomes, where the ribosome will produce specific proteins depending on what's being coded for. So the rough endoplasmic reticulum is going to have proteins produced on its ribosomes, and then those ribosomes are going to be pushed to the inside of the rough ER, where they're going to be modified. From there, once their modification is completed, they'll be shipped to other locations within the endomembrane system. They might be sent to the cell surface, or they might be sent to the cell surface for export. The smooth endoplasmic reticulum is for the production of lipids, which are fats, phospholipids, such as those in our phospholipid bilayer in the plasma membrane, and steroids. So remember, cholesterol is used as a precursor for our sex hormones, estrogen and testosterone. And then there are other steroids that are going to be produced or modified in the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum gets its name because it has no ribosomes on the surface. So there's no proteins that are going to be produced in the smooth ER. The smooth ER is also important in detoxifying the cell, so breaking down things like alcohol, drugs, and other metabolic wastes. Just like cells accumulate mitochondria because they need more energy, cells that are doing a lot of detox are going to have a higher proportion of smooth ER. So for example, liver cells are important in detoxifying alcohol, the system of alcohol and drugs and things like that. And so liver cells are going to have a lot more smooth ER than some other cells. So here's a question for you. How can long-term use of one drug increase your resistance to another different drug that you've never encountered? So this is really important in kind of chronic exposure. This can be anything from antibiotics to heroin. So this can be over-the-counter medication, this can be illegal drugs. But what happens is, as your cells have to detox more, they're going to produce more smooth ER and the associated detox enzymes that the cell needs. Now, the major place for this, of course, is the liver. But with more smooth ER, it allows your body to process these toxins much more quickly. So there you get your increase in tolerance. So the body can break down the drugs you're introducing to your system much more quickly. And that then requires a higher dose of whatever your drug to get the desired effect. Now remember, this could be antibiotics, this could be heroin. It doesn't matter. Your cells will still accumulate more smooth ER, and so there's sort of a blanket or overall decrease in sensitivity. Our last piece of the endomembrane system is the Golgi apparatus. So the Golgi apparatus functions as sort of a shipping and receiving center. So once your rough and smooth ER produce their proteins, their lipids, and their other molecules, they are actually sent to the Golgi apparatus. So here we have these transport vesicles. So here we keep seeing vesicles. Remember, those are just little membrane pockets that are transport vehicles that move along our cytoskeleton. So I think of them as delivery trucks. So they come from the smooth or the rough ER. They bud off, because remember, those are membranes. So they bud off of those other membranes, and they are moved to the Golgi apparatus. Once at the Golgi apparatus, they fuse with it, and then their contents are sort of shuttled through and processed as they go, and then released out the other side. So if we look over here, we've got the side that's closest to the ER. Molecules will enter on that side, sort of shuttle through, and are released out the sides, or depending on the actual structure, maybe even the far end. So once they are finally and completely processed through the Golgi, another transport vesicle is going to bud off, and that is going to be moved to wherever that inside product is needed. So it might be needed in the cell, in which case it will be taken to whatever other organelle or location it's needed, or it might go and fuse with the plasma membrane because it's a protein that needs to be embedded there, or it's a hormone that needs to be secreted. All right, so here's sort of an overview of the endomembrane system. So here we've got some probably proteins being produced in the ER. They're packaged into a transport vesicle where they fuse with the Golgi apparatus. Likewise, we could have lipids or steroids produced in the smooth ER, fuse with the Golgi where the contents are dumped in, and then passed through the Golgi apparatus where they bud off again as a transport vesicle, and in this case, fusing with the cell. Now, once that happens, they're just sort of dumped out into the cell and then can move along in the fluid around the cells and be taken to other cells where they're identified by different receptors and brought in. So all of those organelles that we've talked about so far are present in both plants and animals. But there are a few that are found in only one. So let's talk about one of those, and that is the plant cell wall. Now, our bodies have bones to give us structure. Plants don't have bones. Instead, they have the cell wall, which is a really thick, almost rope-like fiber, or almost, I like to think of it as like a burlap fiber, but it's really strong, and that gives it rigidity and strength because the plant has to stand up against the forces of gravity. So the cell walls help to contribute to the structural strength of the plant. It also gives the plant protection from water loss. It gives it protection from insects and other animals that might eat the plants, because plants can't move. We know that, right? So if something's bothering it, it has to have protection already built in. So if something's bothering you, you swat it away or you move away from it, but the plant doesn't have that option. There are sort of channels in the plant cell wall that allow plants to move water and other molecules in between them. So it's not completely rigid. They can actually communicate. They can move nutrients in between them. Also found within the plant cell is something called a vacuole. So a vacuole is basically a storage unit, and in general are only found in plants, although there are a few instances where they are found in animals. But in a plant, the largest structure actually is something called the central vacuole. So the central vacuole in a plant can store nutrients, it can store waste products, it can accumulate poisons. It can also act as a sort of a pigment reservoir. So that would be used to attract birds or insects that are going to be helping the plant with pollination. And it also helps provide some physical support. So the other function of the central vacuole is to basically fill up with water and keep the plant cells nice and plump. So when your plant looks all wilty, it's because there's not enough water in that central vacuole. But there are different vacuoles for different things. It just really depends on what the specific cell needs. So for nutrient storage, this might be ions or vitamins that the cell needs. We've got waste management, and then, as I said, storing of toxins or pigments for deterring or attracting organisms, and then, of course, physical support. All right, so chloroplasts. Chloroplasts are basically the solar power units in a cell. So chloroplasts are really important because they are the site where energy is harnessed from the sun. So all energy on Earth is harvested in chloroplasts, the chloroplast works to absorb the sunlight energy and convert it into a chemical energy that's going to be used in the cell. So all plants, not all plants, anywhere that you see green on a plant, it's green because of the presence of chloroplasts. So energy is harnessed in the chloroplast and then can be utilized via the mitochondria. So remember, chloroplasts were our other organelle that are believed to have arisen by endosymbiosis. Remember that we said they have DNA and they're similar in size to bacteria, to some bacteria. So the thought is that they were a photosynthetic bacteria that built up a symbiotic relationship with the prokaryote that consumed them and eventually became an important part of that organism. So these little structures here are called thylakoid. These are the light harvesting molecules, and these are the site of photosynthetic reactions, the actual reactions that take that sunlight energy and convert it into chemical energy. So our book has this nice sort of summary. I'm not going to go through all of this. It just gives a really brief description of each of the major organelles and then tells you whether it's in the plant or the animal cell. I would strongly encourage you to make flashcards for these. Take some notes on which molecules are associated, which ones work together, and sort of understand the flow of information, whether it's genetic information to proteins. We of course have our genetic information housed in the nucleus, and then some of that information is sent to the ribosomes on the rough ER where they're turned into proteins, processed through the rough ER and the Golgi, and then eventually excreted out the cell. And then of course we have, or used within the cell. Then of course we have the mitochondria and the chloroplasts, which are our energy converting organelles that have come about via endocytosis.